Johann Gregor Mendel, 1822–1884. Gregor Mendel, through his work on pea plants, discovered the fundamental laws of inheritance. He deduced that genes come in pairs and are inherited as distinct units, one from each parent. Mendel tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. He recognized the mathematical patterns of inheritance from one generation to the next. Mendel's laws of heredity are usually stated as 1. The law of segregation. Each inherited trait is defined by a gene pair. Parental genes are randomly separated to the sex cells so that sex cells contain only one gene of the pair. Offspring therefore inherit one genetic allele from each parent when sex cells unite in fertilization. Point 2. The law of independent assortment. Genes for different traits are sorted separately from one another so that the inheritance of one trait is not dependent on the inheritance of another. 3. The law of dominance. An organism with alternate forms of a gene will express the form that is dominant. Walter Sutton and Theodore Boveri. The Boveri-Sutton chromosome theory, also known as the chromosome theory of inheritance or the Sutton-Boveri theory, is a fundamental unifying theory of genetics which identifies chromosomes as the carriers of genetic material. It correctly explains the mechanism underlying the laws of Mendelian inheritance by identifying chromosomes with the paired factors particles required by Mendel's laws. It also states that chromosomes are linear structures with genes located at specific sites called loci along them. It states simply that chromosomes, which are seen in all dividing cells and pass from one generation to the next, are the basis for all genetic inheritance. Over a period of time, random mutation creates changes in the DNA sequence of a gene. Genes are located on chromosomes. Nettie Stevens. Nettie Stevens and Edmund Beecher Wilson independently developed the idea of sex determination by chromosomes. Their work established the importance of chromosomes in heredity and helped Thomas Hunt Morgan interpret the early genetic results from Drosophila. In 1903, Stevens got her PhD from Bryn Mawr and started looking for a research position. She was eventually given an assistantship by the Carnegie Institute after glowing recommendations from Thomas Hunt Morgan, Edmund Wilson and M. Carrie Thomas, the president of Bryn Mawr. Her work on sex determination was published as a Carnegie Institute report in 1905. In this first study she looked at sex determination in mealworms. Later, she studied sex determination in many different species of insects. Stevens' assistantship at Bryn Mawr still meant that she had to teach. She wanted a pure research position, and wrote to Charles Davenport to see if it was possible for her to work at his station for experimental biology. Unfortunately, Stevens died of breast cancer in 1912 before she could occupy the research professorship created for her at Bryn Mawr, or work with Davenport at Cold Spring Harbor. Thomas Hunt Morgan. Morgan was curious as to why female flies never had white eyes, and he considered several possible reasons for this phenomenon. One potential explanation was that white-eyed females never hatched, or that they died early in development. In other words, this hypothesis predicted that white eyes were lethal in female flies. Therefore, among the progeny of a test cross of heterozygous F1 red-eyed females to white-eyed males, there should be no white-eyed females. Morgan conducted this very cross to see whether the results matched his predictions. Surprisingly, this cross yielded a 1:1:1:1 ratio of red-eyed females to white-eyed females to red-eyed males to white-eyed males. Based on these results, Morgan arrived at three important conclusions. The appearance of white eyes in females shows that this trait is not lethal in females. All possible combinations of white eyes and sex are possible. The white-eyed trait can be carried over to females when F1 females are crossed with white-eyed males. Beadle and Tatum. The one gene, one enzyme hypothesis is the idea that genes act through the production of enzymes, with each gene responsible for producing a single enzyme that in turn affects a single step in a metabolic pathway. The concept was proposed by George Beadle and Edward Tatum in an influential 1941 paper on genetic mutations in the mold Neurospora crassa, and subsequently was dubbed the one gene, one enzyme hypothesis by their collaborator Norman Horowitz. In 2004, Norman Horowitz reminisced that these experiments founded the science of what Beadle and Tatum called biochemical genetics. In actuality, they proved to be the opening gun in what became molecular genetics and all the developments that have followed from that. The development of the one gene, one enzyme hypothesis is often considered the first significant result in what came to be called molecular biology. Although it has been extremely influential, the hypothesis was recognized soon after its proposal to be an oversimplification. Even the subsequent reformulation of the one gene, one polypeptide hypothesis is now considered too simple to describe the relationship between genes and proteins. Oswald Avery. The Avery-McLeod-McCarty experiment was an experimental demonstration, reported in 1944 by Oswald Avery, Colin McLeod, and Maclyn McCarty, that DNA is the substance that causes bacterial transformation, in an era when it had been widely believed that it was proteins that served the function of carrying genetic information, with the very word protein itself coined to indicate a belief that its function was primary. It was the culmination of research in the 1930s and early 20th century at the Rockefeller Institute for Medical Research to purify and characterize the transforming principle, responsible for the transformation phenomenon first described in Griffith's experiment of 1928 killed Streptococcus pneumoniae of the virulent strain type 3S, when injected along with living but non-virulent type 2R pneumococci, resulted in a deadly infection of type 3S pneumococci. In their paper, Studies on the Chemical Nature of the Substance-Inducing Transformation of Pneumococcal Types, Induction of Transformation by a Desoxyribonucleic Acid Fraction Isolated from Pneumococcus Type 3, published in the February 1944 issue of the Journal of Experimental Medicine, Avery and his colleagues suggest that DNA, rather than protein as widely believed at the time, may be the hereditary material of bacteria, and could be analogous to genes and/or viruses in higher organisms. Rosalind Franklin and Maurice Wilkins. Rosalind Elsie Franklin, the 25th of July 1920, the 16th of April 1958, was an English chemist and X-ray crystallographer whose work was central to the understanding of the molecular structures of DNA, deoxyribonucleic acid, RNA, ribonucleic acid, viruses, coal, and graphite. Although her works on coal and viruses were appreciated in her lifetime, her contributions to the discovery of the structure of DNA were largely recognized posthumously. Maurice Hugh Frederick Wilkins CBEFRS, the 15th of December 1916, the 5th of October 2004, was a New Zealand-born British physicist and molecular biologist, and Nobel laureate whose research contributed to the scientific understanding of phosphorescence, isotope separation, optical microscopy and X-ray diffraction, and to the development of radar. He is best known for his work at King's College London on the structure of DNA. James Watson and Francis Crick. Deoxyribonucleic acid, DNA, is a double-stranded, helical molecule. It consists of two sugar phosphate backbones on the outside, held together by hydrogen bonds between pairs of nitrogenous bases on the inside. The bases are of four types: A, C, G, and T. Pairing always occurs between A and T, and C and G. James Watson, 1928, and Francis Crick, 1916-2004, realized that these pairing rules meant that either strand contained all the information necessary to make a new copy of the entire molecule, and that the order of bases might provide a genetic code. Watson and Crick shared the Nobel Prize in 1962 for their discovery, along with Maurice Wilkins, 1916-2004, who had produced a large body of crystallographic data supporting the mode. Working in the same lab, Rosalind Franklin, 1920-1958, had earlier produced the first clear crystallographic evidence for a helical structure. Crick went on to do fundamental work in molecular biology and neurobiology. Watson became director of the Cold Spring Harbor Laboratory, and headed up the Human Genome Project in the 1990s. Jacob Monod. In 1966, the Jacob Monod model for gene expression describes the very first genetic regulatory mechanism to be understood clearly. When it was first described by French biologist François Jacob and Jacques Monod, who originated the idea that the control of enzyme levels in cells occurs through the regulation of transcription. And they, along with another scientist, shared the 1965 Nobel Prize in Medicine for their work on what is called the lac operon. The protein that is formed by the lac gene is known as the lac repressor. It has two types. They are positive regulator and negative regulator. Barbara McClintock. For much of the 20th century, genes were considered to be stable entities arranged in an orderly linear pattern on chromosomes, like beads on a string. In the late 1960s, Barbara McClintock challenged existing concepts of what genes were capable of when she discovered that some genes could be mobile. Her studies of chromosome breakage in maize led her to discover a chromosome-breaking locus that could change its position within a chromosome. McClintock went on to discover other such mobile elements, now known as transposons. She also found that depending on where they inserted into a chromosome, these mobile elements could reversibly alter the expression of other genes. Johann Gregor Mendel, 1822–1884. Gregor Mendel, through his work on pea plants, discovered the fundamental laws of inheritance. He deduced that genes come in pairs and are inherited as distinct units, one from each parent. Mendel tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. He recognized the mathematical patterns of inheritance from one generation to the next. Mendel's laws of heredity are usually stated as 1. The law of segregation. Each inherited trait is defined by a gene pair. Parental genes are randomly separated to the sex cells so that sex cells contain only one gene of the pair. Offspring therefore inherit one genetic allele from each parent when sex cells unite in fertilization. Point 2. The law of independent assortment. Genes for different traits are sorted separately from one another so that the inheritance of one trait is not dependent on the inheritance of another. 3. The law of dominance. An organism with alternate forms of a gene will express the form that is dominant. Maxim Gilbert. Maxim Gilbert sequencing is a method of DNA sequencing developed by Alan Maxim and Walter Gilbert in 1976–1977. This method is based on nucleobase specific partial chemical modification of DNA and subsequent cleavage of the DNA backbone at sites adjacent to Maxim Gilbert. This sequencing is a method of DNA sequencing developed by Alan Maxim's modified nucleotides. His sequencing was the first widely adopted method for DNA sequencing and, along with the Sanger-Dideoxy method, represents the first generation of DNA sequencing methods. His sequencing is no longer in widespread use, having been supplanted by next-generation sequencing methods. First transgenic animal. In 1974, Rudolf Janisch created a transgenic mouse by introducing foreign DNA into its embryo, making it the world's first transgenic animal. However, it took another 8 years before transgenic mice were developed that passed that transgene to their offspring. Genetically modified mouse has its genome altered through the use of genetic engineering techniques. Genetically modified mice are commonly used for research or as animal models of human diseases, and are also used for research on genes. Kary Mullis. In 1985, Kary Mullis invented the polymerase chain reaction, PCR, a method of amplifying or producing many copies of a specific piece of DNA. The revelation came to this eccentric character on a drive in Northern California. Source: DNAi. Kary Mullis explains how the polymerase chain reaction, PCR, was named. PCR is a method widely used in molecular biology to make many copies of a specific DNA segment. In the first step of PCR, the two strands of the DNA double helix are physically separated at a high temperature in a process called DNA melting. First bacterial gene sequencial, Haemophilus influenza. Haemophilus influenzae is a gram-negative bacterium capable of causing respiratory tract infections as well as severe invasive disease. This species is characterized by the presence, absence of a polysaccharide capsule. 1. In the present era of conjugate vaccines against Haemophilus influenzae type B, non-vaccine preventable strains are of concern. Here, we report the first whole genome sequence of an invasive H. influenzae type B strain. This genomic information will enable further investigations on encapsulated non-type B H. influenzae strains. Ian Wilmot. Sir Ian Wilmot, born July 7, 1944, Hampton Lucy, Warwickshire, Eng, British developmental biologist who was the first to use nuclear transfer of differentiated adult cells to generate a mammalian clone, a Finn Dorset sheep named Dolly, born in 1996. In 1995 and 1996, researchers at the Roslin Institute in Edinburgh, Scotland, cloned mammals for the first time. Keith Campbell, Jim McQuirr, William Ritchie, and Ian Wilmot cloned 2 sheep, Megan and Morag, using sheep embryo cells. The experiments indicated how to reprogram nuclei from differentiated cells to produce live offspring, and that a single population of differentiated cells could produce multiple offspring. They reported their results in the article, Sheep Cloned by Nuclear Transfer from a Cultured Cell Line, in March 1996. This experiment led the Roslin team to later clone mammals from adult body cells and to genetically engineer